a week from today, regrade requests will be due. Written electronically, preferably by email to me. Make sure you've looked at the keys first, tried to figure out which questions you missed. Send me, if you think that a question was graded incorrectly compared to what the key says. Send a request. Right. Any questions before we get going? Well, we've been going, I suppose. Anything? Okay. So the brief one-slide review. Last class we talked a bit about reporter genes, among other things. And I just wanted to remind everybody that the setup for this experiment in yeast was that we had different TATA boxes. Those are the elements that TATA binding protein binds. There's either TA, TA, or TG, TA. And those are in the promoters upstream of the transcription start site for what gene? This is the reporter gene. This was LAC-Z, which is the gene that encodes beta-galactosidase, which is the enzyme that converts this substrate on the left into the colored substrate, a yellow-colored substrate product on the right. So where does, I heard some of you say TATA binding protein was the gene. So how does TATA binding protein fit into all of this? Back to transcription regulation, trying to wrap it up today. Yeah.
Because you have to recognize that sequence in order to tell where the transcription will start.
Right. So TATA binding protein is going to bind TA-TA presumably and not TG-TA. So the prediction is that when TATA binding protein binds the genome, right, this circular molecule is a plasmid. It's a circular molecule of DNA that we inject or electroporate, use electric shock to get it inside the yeast. It works. I don't know who figured that out, but it works. And that plasmid carries either of the two TATA sites, TA-TA or TG-TA. TATA binding protein will bind TA-TA and not TG-TA. So for the top case, TA-TA, we'll get a lot of transcription, we'll get a lot of yellow product produced. If you provide a normal TATA binding protein with the mutant version of its DNA sequence that it recognizes, you get no transcriptions, you get no beta-galactosidase produced, you get no yellow product. So that's, in a nutshell, what we talked about last class. And that is the same LAC-Z that we talked about when we talked about the lac-operon in bacteria and the LAC-ZY and A genes. That is LAC-Z, the beta-galactosidase. So now that we're all back up to speed, I want to show you some of your responses. You might recall that at the end of last class, I asked everybody to pick one of these 4 genetic pathways shown on the right, and just pick one, and to draw how the protein levels or mRNA levels, gene expression levels of A and B change over time given their relationships, 1, 2, 3, or 4. So I'm showing you some anonymous feedback, suggestions that you made about these pathways. So this is pathway 1. Which one of those two do you think reflects the relationship between gene A and gene B? Which one's better? Is it the, yeah, these are both responses I got to pathway 1. Is it the top one or the bottom one? Okay, so somebody explain how we come up with a graph like the one on the top. So I said the initial state was what about gene A? It was present. So gene A is present, or the protein is at time 0. By the way, these axes were supposed to be labeled. So that's time. From time 0 to time, who knows, infinite maybe. So we have A present at time 0. So then what happens after that?
It starts turning into B.
Okay,
Well, not turning
Well,
into
no, not turning B, it activates something that triggers B.
I'm glad you said turns into B, because that's a point of confusion I want to make sure is as clear as possible. That these diagrams on the left don't, for example, indicate that A is becoming B. This is a genetic pathway. This is not like a biochemical pathway like we saw in the previous slide, where ONPG gets cut by an enzyme and turns into ONP, the yellow-colored chemical, and a sugar. This is gene A and how it regulates or controls another gene. So in this case, gene A activates or turns on the activity of gene B. So in this case, we're saying turning on or regulating activity means it's causing the expression, the production of protein from gene B. So my only question, I don't know, maybe the student that submitted the top version is in the room now. Maybe they want to fess up to having drawn this one, or maybe not. I want to know why this happens to gene A protein levels over time. And if it wasn't you, do you have a theory?
This should just be linear, right? I mean,
So another possibility is that.
it can be exponential.
So if you put either of those down on the drawings that you made for this exercise, both would be fine. What's the difference between what happens to protein A in line 1 and line 2? What's happening?
It stays constant on line 1. It's
Right.
present though at all times.
It's present and it doesn't go away. Right? The same amount of protein A that you started with is just present in the same amount the whole way through the experiment. So in contrast, what's happening for line 2?
It's decreasing as B becomes more.
So why is it decreasing?
It's being used up as for in a pathway.
Okay, so this again gets back partly to that hopefully not too confusing point about that this is not gene A becoming gene B, because this is not a chemical reaction, this is one gene turning on the production of another. So what's hopefully being represented here, and this is actually, the second line is actually what I drew when I was drawing out the key that I thought I would present to you, but I decided it would be better if you came up with the answer key. That we know that over time, both messenger RNAs and proteins get degraded by the cell. So if you're not making any new protein in a cell, then over time, the amount that you started with will get slower and lower and lower as every molecule of that protein eventually gets degraded, broken up by the cell, and recycled into new proteins. That's what our cells do. We take existing proteins in the cell and new nutrients as well, but some proteins get recycled by the cell and turned into amino acids and incorporated into new proteins. I wanted to show this bottom example because, wow, I do not want the highlighter, sorry. This is not A becomes B. So that's the point of showing that bottom one. That's like a chemical reaction. This is a biological control of one gene by another. And if you want to talk about that in more detail, I definitely urge you to come talk to me after class, in office hours, something starting next week, because this is an important concept, and I want to make sure I've got time one-on-one with you if you want to discuss concerns. So then you might have expected that, yes, I would show you an example of pathway 2 next, and then this next slide is pathway 3, and the slide after that's pathway 4. I didn't get many submissions for this one. I got a lot of number 1s. Maybe people thought that was the easiest. I didn't get very many submissions for pathway 3 either. It's complicated looking. But is this accurate? Is this what would happen if you had pathway 2 going on? Just have an initial amount of protein A, gene A expression. It's constant, or based on the previous slide, what else could we draw this? Yeah, and to be honest, it doesn't matter to me if you show it doing anything like that. As long as you know what you're doing and you can justify it, that you can say, we start with a certain amount of protein, it slowly degrades over time. So the amount of protein A we have gets lower and lower as time goes on. That's fine. Right. We'd have to talk about biochemistry and the kinetics of decay for you to figure out what the mathematical description of that decay is. So I'm not worried about the shape of the line. So it could stay constant, it could decrease. But what happens to B? That's the essence of all of these questions. What's the effect of gene A on gene B? So gene A suppresses gene B in this case. And if gene A is present at the start of time, gene B never gets made. So I believe, having not spoken with this student, that's why there's a B down here at 0 on the y-axis, and then you don't see anything. So if gene A is present, and if gene A prevents the expression of gene B, then you're never going to see any gene B. Perfect. This is a good answer.
But wouldn't gene B be created once A degrades?
Well, it depends on how gene B gets turned on, how the expression of gene B gets turned on. I'd have to add a third letter in here. So we could do that. We could say C activates B. And so as long as A is there, B gets suppressed. But if there's some gene C, a constant level, say, then if this is what happens to the amount of gene A over time, what would we draw in terms of B?
It would increase over time.
Right, C is always there. So C is always able to turn on expression of B, but we have to wait for A to get the hell out of there.
So is that intersection there like a threshold?
Well, again, the intersection is sort of meaningless because I haven't told you about the kinetics of the interaction between genes A, B, and C. So where the lines cross is not important in this case. It's just the direction of the lines that I care about. Are they going up, are they going down, or are they not changing? Yeah?
So on a test, would it be more accurate to draw like as A reaches 0 and then the B line would start off like a B?
That would be more accurate, but I would be super impressed and happy if I saw that, but it's not something I would expect. And again, it's because I haven't told you exactly what the relationship is between gene A and gene B. So what I hear you saying, if I interpret correctly, is we have to wait until every last molecule of gene A is gone before we start seeing an increase in the production of gene B. So that would be a very literal and accurate interpretation of what could be going on. That would be fine. So in this case, this number 2 response was an accurate response to this question. We've got 2 different submissions here for pathway 3. That top one might be a little bit hard to see, so I'll blow it up a little bit. Like that. So think about it for a minute, and then we're going to take a quick vote on which one of those 2, the top or the bottom one, is probably more accurate. B represses A, A activates B. Over here. I'm kind of inspired to make this sort of thing like an exam question. Here's a pathway. Here are 4 possible plots of their interactions. Which one is most accurate? Right. And good, I recorded that, so I'll remember to use it.
I would think the top one would be more accurate, but it's only like half of the process.
Yes.
Okay, so the top one's more accurate, but it only shows half of the process. Well, the good news is, both of them start with A in abundance, which is what the problem said. So at least we've started off well. Both of them have B absent. So from there, what's the difference?
One of them shows A decreasing as B increases, the other doesn't.
Right. So it's almost like, if I understood your initial comment correctly, it's kind of like we're looking at the same part of the
Except that
plots.
A
We're just looking at,
shows decreasing immediately in the second one.
right. And that depends entirely, so you have no way of knowing what A is going to do. That's what we talked about in the first example. You're free to choose, do you think A, the protein level of A stays constant over time, or do you think it decreases? Because the protein degrades, for example. Either of those is fine. So in both of these examples, B increases. Does that make sense? Why does B increase, the amount of protein B? Why? Right. Gene A activates gene B. So we have A present initially, so as soon as we start time, there's going to be the production of gene B. So in both of these plots, we see the amount of B increase. So then, what about the other arrow in this process? B inhibits A. What should that do to the plot? So this is, I think, where we get the difference between the top submission and the bottom submission. Is in one, B increases and then just stays there. But A doesn't change. So there's a fundamental question about this interaction between B and A that I didn't explain and that might explain the difference between these two submissions. I'm willing to say both of those are accurate, but as long as you know, if you created those, as long as you know why you drew what you drew. So here's my explanation, just in case. What does it mean when B represses A? Okay, we've got two, I think what's happening here is we have two interpretations. We have no more A production. So which plot would that be? Okay, what's the other explanation for B, for what's happening in the cell when we write B represses A?
Repressing activity.
Pardon?
Repressing its activity.
So maybe more than that. You could have, it's sort of like a positive and a neutral and a negative. This top one's sort of neutral. A represses B, or sorry, B represses A, that means you get no more A made. That's what I was thinking when I drew that. That's sort of neutral. It's like you get to keep the protein A that you started with, you just don't make any more. What's the other possible explanation?
Can you stop production of B, gene B as well, because you don't have no more A?
Right. So if B represses, this is important. If B is preventing A from working and A is important to get B, then there's going to be some sort of equilibrium that's reached. Yeah?
It's like a loop, right? So if one represses the other one and the other activates, then nothing would happen at the end.
This is a feedback.
Exactly the same.
This is a feedback loop. And that's why I wanted to put this in, because we're about to look at a real example of a feedback loop, not just a made-up one. So the other possible interpretation of this pathway, B represses A, is that B actively gets rid of A. And I think that would predict that, the one on the bottom, that as you make B, you lose A. The amount of A actually goes down. But if you have B represses the production of A, then you might get something that's more like the top plot, where you start with a certain amount of protein A, and you just don't make any more. Yeah.
So if we're talking about the top one as being target, would they both be flattened out eventually, so no more B can be made? Or if we're doing the bottom ones, where it's deteriorating it, wouldn't B go down to 0 again eventually?
If all proteins degrade over time, then sure. If we looked over more time, the amount of B would go down to 0. So bottom line, if we've lost you at this point, perk your ears back up. And remember that all I ask you to do if you face this in real life, in the laboratory, or on an exam, is that you be able to explain why you're drawing the lines that you draw. So any decrease in protein amount could be 2 reasons. One is that the cell is just degrading the protein. The other one is that one gene is actively degrading the other protein. Yeah.
So, assuming that, we're talking about the top one where B does not eliminate A.
Right.
Wouldn't they naturally kind of degrade over time themselves anyway?
Right. So if we look infinitely into the future, everything goes to 0. And that would be fine. If you decide to draw the most accurate version of any plot possible. No matter what's happening. If A, for example, this would be that pathway. A positively interacts with the gene B, or gene A stimulates the production of gene B. Then you'd get that. The gene B would increase to a certain level. It doesn't have to be the same level as A. That's arbitrary. Gene B could go up to that level. And then over time, by infinite time, if at some point you turn off transcription, which presumably would happen, say the organism dies, protein levels go down to 0. Yeah.
Why does it remain constant even though it's activating gene B?
Why does it or why doesn't it?
Why does it remain constant? Like, what do we assume about?
So the assumption, right, so that's a good point. Should I draw A as never being a plateau? Should A always be sort of constantly decreasing at a certain rate?
After a certain time of day it's possible because
I'd be happy with that. So A could always slowly be decreasing because I haven't told you how A gets made. What if this happens?
Then it'll stay constant.
you get small oscillations.
This is a feed-forward loop. A turns on B, B turns on A turns on B, B turns on A. Then you'd get something that, yeah, A is always present, B is always present. And honestly, although these are sort of made up, this is how organisms regulate gene expression. All of these different pathways exist in cells between different genes, not just gene A and gene B, but genes that have real names and real functions. This is how patterns of gene expression get produced by cells, how some genes turn on other genes, some genes turn off the production of genes. That's how our cells regulate our genes, so that we're not making all the proteins for all 30,000 of our genes all of the time in every cell. What a waste. If my toe cell was making the proteins that my eyes need to absorb light and see. Right? This is how that works. Yep. Interactions between genes, some genes turn off and keep off the expression of others. Like in this case. This is an accurate response. We start with some gene A. This student, I think, maybe is hypothesizing we start with a little bit of gene B. That's fine. What happens in pathway 4? A prevents B from being made. B prevents A from being made.
Assuming they're in equal amounts, it should relatively take.
Yeah, if they're in equal amounts, then you'd expect this sort of a disaster to happen. nobody gets produced.
Dr.
Yeah,
If we're starting with just A and no B, and A prevents B, wouldn't A just be steady the whole time?
After a while. I mean, you get a little bit of, like a dip from whatever B was present, and then you have a steady stream.
so you could, those in blue would also be reasonable answers to this. You start with A always prevents B, no B ever gets made, so there's never any B here that would do this repression event. That's fine. So either of those is fine. This student just took the liberty of saying, okay, I'm going to start with a little bit of gene B. And that little bit of gene B presumably is enough to start the decrease in amount of protein A. Either is acceptable. So let's wrap up by going back to looking at sleep patterns. Since it's about the weekend, many of you may be staying up late tonight. I know I will not, but that's okay. So those of us that have normal sleep-wake patterns, or not, we know that those, based on our presentation from Monday, we know those are based somewhat on mutations in the cry1 gene, which again doesn't stand for crying. And one of the things we heard about in that presentation was the genetic pathway between these different proteins. So there's a protein that's cry. It represses transcription of a gene called clock. And the relationship between clock and cry is that clock turns on the transcription of cry. The only other thing I need to tell you before I set you loose to draw this plot, this is the information I should have given you on these crazy made-up exercises we just talked about. Clock is a transcription factor. We heard about this on Monday. So it's a gene that goes around and turns on the transcription of other genes. Some of the genes that it turns on are genes because it's named clock. Genes that actually have circadian expression patterns. Genes that are involved in regulating our sleep-wake cycles. The actual genes that do the job of making sure that we don't go crazy. And it also turns on cry. Cry is a repressor. How would a repressor work? Molecularly, inside the cell, what would the protein do you think? So what does the repressor protein do? It blocks transcription by binding to, if this is a prokaryote, an operator. I sound quite British with the operator. Exactly. So think of cry as a protein that will go just bind DNA in the promoter of clock and make sure that clock doesn't get transcribed. So cry is repressing clock transcription. And clock then turns on transcription of cry. So with those details in hand, take 5 minutes or so, or less, and do exactly the same plot as the ones that you did last class and we just talked about. What's going to happen over time between the levels of cry and clock? And let's assume that we start with the cry protein being present.
Are we starting with any clock protein?
No. Or let's say a little bit. Let's see what happens. The one thing that I've learned in many decades as a scientist, not true, many years as a scientist, is that there's never an absolute. So there's always a little bit of everything. Safe assumption. Very few absolutes in science. So yeah, go ahead and work on this. What's going to happen over time in this pathway?
I'm assuming there's some other clock, right?
I know, like, cry suppresses the transcription. So if cry is there, that can't be transcribed. So what I think is cry will be, since cry is there, also if you assume they don't degrade over time. And I feel like
Right. So it'll go, just like 50%. So I think it'll kind of like that. Because you still clock your cryo, but it's going to go ahead and produce your clock. So right now, while we're in but then it's going to right, then you start going on that down.
Okay. So what's going to happen at the very first, at time equals 0 plus something infinitely small? What's going to happen to the initial state?
Cry will increase a little.
So there's some clock present, which means you're going to make a little bit of cry. And all that cry protein that's present is going to do what? It's going to keep clock from being transcribed. So maybe it stays steady, maybe it goes down a little bit. Okay. But then what happens? So this is kind of a steady state. We've got a lot of cry, we've got a little bit or no clock. But what did we just discuss happens to every protein over time? It degrades. So cry starts going down. And as the amount of cry starts to decrease, clock starts getting produced.
Isn't there so much cry that it would get rid of all the clock?
Sure, so it gets rid of all the clock down here. But as that cry protein starts being reduced here, then clock is able to start being synthesized again. But then as clock gets made more and more, what happens? It produces more cry, and what winds up happening is you get an oscillation. And that's just because one protein represses the other, but you have to couple that with this idea that over time any protein degrades. That's the only way to get this oscillation. And it turns out that's exactly what happens in this movie that we started this with. If you remember, we get oscillation. the amount of one protein goes up, but this other protein turning on turns the other protein off. They flip-flop, they go back and forth, and this is the transcriptional control of circadian rhythm. What we heard about on Monday was in humans, this is what happens in cyanobacteria. It's the same process. It's just a simple feedback loop. One protein turns one on, that protein turns the other one off. This is what results. That's it for Friday. Have a fantastic weekend, and I'll see you on Monday for a new topic.